First of all, for those that don't know me, I'm Andrew Heaton, CSO at the CureJM Foundation. Been with the foundation for around about 5 wonderful years so far. So it's my very great pleasure to welcome everybody on behalf of the board of the CureJM Foundation, staff from the foundation, and obviously all our families and patients, which is what we're all here to serve. The foundation's just completed its 20th year of operations. We're funding a wide variety of programs, and it seems to expand every year. This covers basic research, genetics, translational programs, clinical initiatives, diagnostic initiatives, mental health initiatives. We really span the full gambit. We're also active in funding our clinical care network and our centres of excellence. The catalyst for this meeting is really the staff are in the foundation are in the very fortunate circumstances. We interact very much on a daily basis with so many leaders in our different clinics. And I think the real catalyst behind this was Dr. Susan Kim, who's presenting today, gave a wonderful presentation to our research committee several weeks ago. And we were all blown away with the work that she was doing with her EPIC tracker. And we thought, wow, I wonder how many other people in our network are aware of what is going on. And in talking to so many of our other clinicians that are doing great work, we thought it would be a wonderful opportunity to actually gather our broader clinical network together and share with you from an educational perspective what some of our leaders are doing in their different clinics, but also perhaps open up new pathways for collaboration. The foundation believes that 2024 is going to be a breakout year, even though last year was our 20th anniversary. I think 2024 holds promise for some significant milestones. We know that there have been several things that have been boiling along with some of our centres for the last few years that are about to come to fruition with some potentially fantastic clinical trials in JM patients. I think, as we all know, because there are no medications registered for JM, we are all keen to see, what we can do as far as advancing clinical trials to actually get indications for some of our more important drugs. The other things that are going to happen in 2024, we've got a range of important meetings. These include GCOM in March. We hope to catch up with everybody at GCOM. I know several of the CureJM staff will be going to that. The foundation, in collaboration with Duke, particularly Dr. Jeff DeVerchten, will be running our normal annual virtual medical symposium in around about May or June. So after GCOM, we're going to run a second of these clinical care networks / COE educational symposia later in the year, sometime around September. So we'll be looking for volunteers to present at that. The other things the foundation will be doing in the not too distant future is our large in-person meeting that so many of you would have attended. The next scheduled one is in Chicago in 28th of June 2025. So again, another really important meeting that's coming up for us. During this year, we've got several chapter events, and also we have a series of small satellite meetings. These include one in Texas coming up and another in New York. If you contact Lauren, she'll be able to fill you in on the details with those different meetings. From a housekeeping perspective, firstly, if people could keep themselves on mute while folks are presenting, that will ensure we don't get too much crosstalk, speaking over people. Each presenter will be presenting for somewhere between 10 and 20 minutes. There'll be ample opportunity to ask questions. We would prefer people to post their questions to the actual chat box. The presenters will then answer the questions from the chat box as we go through. So as I said, today's meeting is really about education and collaboration. And I think from a collaborative perspective, one of the most important things we have from a basic research perspective is access to clinical trial samples or samples from patients, both freshly diagnosed and patients that are on different medications that people choose to use in their basic discovery program. So the foundation has funded for the last several years a CARRA JM repository and registry. And it's my very great pleasure to introduce as our first presenter, somebody we've worked with for a long time that is responsible for this important asset that so many of you will use or have used and will use in the future. So we really wanted to remind folks about how important this asset is and try and get some more people to sign up so that we can get more samples, because with a rare disease, one of the key things to furthering research is ensuring we've got a broad range of samples, these critical samples for people to utilize in their further studies. So my very great pleasure to start the meeting off today and introduce Karin Park from CARRA, and she's going to talk to us about the biorepository and registry we have. Karin, over to you.
Thanks so much, Andrew. Let me just share my screen. First, thanks so much for the opportunity for us to present. I think this is a wonderful opportunity and platform for us to share more about the JDM repository and data that we have available. I think certainly a great opportunity, and we are looking forward to talking more about some of the requests that we've received and just progress overall in terms of enrollment and sample collection. So first, I'm Karin Park. I'm the Associate Director of Research Operations at the Childhood Arthritis and Rheumatology Research Alliance, CARRA. I've been at the organization for 3 years now, and in my role primarily focus on operations concerning our registry and biorepository and how that work gets integrated within our organization. So for today's brief presentation, I'll start with a very quick overview of CARRA and the CARRA Registry in case there are attendees who may not be so familiar with our organization, and then talk more about our partnership and, like I had mentioned, some updates with our registry and repository, and then a couple of quick reminders about resources that are available through CARRA to help support efforts to advance JDM research. So first, who we are. CARRA was founded in 2002 with a mission to conduct collaborative research to prevent, treat, and cure pediatric rheumatic diseases. Our work is driven by many of you who I see on this call today, primarily clinicians and researchers, as well as other health professionals and patients and families. We have over 600 members within CARRA, which actually captures most of the pediatric rheumatologists across North America. We'll talk more about our registry, and of course, we have strategic partnerships with organizations like the CARRA Foundation, as well as other disease-specific organizations, as well as industry partners and international collaborators as well. So the CARRA Registry is an observational prospective registry that was launched in 2015, in which we collect both clinical and patient-reported information about patients with pediatric rheumatic diseases, including arthritis, lupus, and dermatomyositis, as well as scleroderma. We have about 70 active registry sites that are primarily US-based, but some sites in Canada as well. And at each of these sites, there are a number of registry site staff who contribute to data entry in our registry, and this is collected about every 6 months per participant. We also have a sample repository with 2 central locations, one in Cincinnati, Ohio, and one in Toronto, Canada. And this was added after we had launched the registry to really bolster the clinical data that we were collecting and support translational research as well. And we aim to follow participants for at least 10 years into their adulthood. So the registry was primarily designed to evaluate long-term safety of medications used in pediatric rheumatology to support post-marketing surveillance studies, as well as other types of research studies. We are also able to study clinical outcomes associated with treatments and clinical course of these diseases that are captured in the registry. We have standard data collection forms and medication logs, which were designed and continue to be refined even today by our registry's scientific directors and in partnership with disease experts through CARRA's research committees. And at the time of the registry launch in 2015, The initial focus of the registry was on JAA only, though we have been able to expand our disease focus over time thanks to strategic partnerships like that with CARRA. So really our formal partnership began many years ago, I think about 6 years ago, in 2017, and focusing on the CARRA Registry and Biorepository and really creating a valuable resource for the JDM community. So at the start of our partnership, really CARRA was just beginning to implement JDM data collection and still onboarding registry sites and introducing them to this new cohort. So our protocol was written in a way that is broad and allows sites as they come on to really just get started with enrolling any of the JDM patients that they are seeing at their site. So there's hopefully few barriers to start enrolling for this cohort. And in terms of our goal, we set out with an initial goal to enroll 400 JDM participants, which at that time, 6 years ago, was really a ballpark estimate for this rare disease. But certainly we are committed to enrolling as many potential JDM patients that we see at registry sites. And we also recognize, of course, that business development activities and broader partnership efforts are really important to ensure that the registry is sustainable and also to ensure visibility for this valuable resource. So here I've just listed some of our current JDM and JPM inclusion/exclusion criteria. So notably with this cohort, this is an inception cohort, which I think is quite unique in our community. So notably diagnosis before age 19 and within 6 months of registry enrollment. So we're really able to capture this population early, which with research, of course, is a great benefit. And so this is also a unique characteristic because we don't have inception cohorts necessarily for other diseases captured in the registry. So certainly I think that speaks to the value that we've been able to create for the community. So progress to date. The registry so far, we have over 14,000 participants, most are JA, but we've made some significant progress with enrolling lupus and JDM patients as well. Of note, we've surpassed our goal of 400 JDM patients, and we will continue to enroll participants. And in a slide or two, I'll hone in a little bit more on the JDM participants and annual enrollment stats. For biorepository, we have over 1,700 biosamples collected across the registry. We currently have 11 active biosample cohorts. And something to note is that with each of these cohorts, they are generally tied to specific sub-studies with PIs. So there is typically a grant associated with our cohorts, but that's not the case for this JDM cohort, which actually I think is a unique benefit, since samples really are available to many in the community rather than initially those study investigators and then to the community afterwards. Among the JDM samples, we have about 180 samples in total, and we recently worked with Dr. Susan Kim, our next speaker, to launch another JDM focused sample cohort, which Jump to Act, which focuses specifically on active skin disease in JM. And I also wanted to note that in this graph, you'll see that the JDM biosample cohort is actually one of CARRA's largest biosample cohorts in the network, behind a couple of externally funded studies, which is truly an incredible resource. So honing in on the JDM cohort, this chart shows the cumulative growth of enrolled participants and collected biosamples over time. So on average, just looking at the past 4 years, our average enrollment is about 56 patients a year and about 35 samples collected each year. Of our 70 active sites, 55 sites have enrolled at least one JDM participant. We've seen that about half of the sites, around 29, have between 1 and 4 JDM patients, and the other half 5 or more participants. And so this is a really great testament to the engagement of our registry sites. This is over 80% of the active sites in our network. And generally, we do see that sites with more JDM patients tends to be consistent with sites we know are larger and do see a higher volume of pediatric rheumatology patients. And in terms of sample collection, we have 114 samples collected at baseline or their enrollment visits with the registry, and 65 follow-up visits at the 6-month clinical visit. And most of our sites are able to collect centrifuge or spun samples. So I think this is also really important because with our standard collection kits, that means that we're able to get DNA, RNA, plasma, cells, and serum. There's a small subset of sites that are only able to collect uncentrifuged or unspun kits, but we're still able to get DNA and RNA on those patients. But for the most part, most of the sites are able to centrifuge samples. And I believe that 22 of the 55 sites, about 40%, have collected biosamples, as well as enrolling their participants. So through CARRA, one of the benefits of membership is being able to request data and samples for use. So we do have a process through CARRA to review and approve requests to make sure, obviously, that it's feasible, what folks are looking for in terms of clinical data is collected in the registry. Same thing for samples as well, making sure that folks really have the resources and bandwidth to be able to analyze the samples, and that we're really being mindful that samples are a finite resource. But at the same time, we don't want to just keep all the data and samples within CARRA. We really want to be able to disseminate that to investigators to support research being done in the community. So to date, we've had 24 total data sample requests that leverage JDM data and samples, broken down by status here. And of these, 19 of the requests are using our current registry. So really that's the registry we launched in 2015. Prior to 2015, we had a legacy registry. So some of these 6 completed projects also use legacy registry. But for our purposes, we'll cover some highlights from the current requests using the CARRA registry. Of note, 3 of these requests include use of the JDM samples. So as I mentioned, I really think it is a unique benefit that these samples are not tied to one specific study. They're really available to all members. And so I think really this is the cohort where we have seen actually the most requests to date to use these samples. So I won't go over all of these samples, but certainly can share these after the deck so folks can get a better idea of projects going on through CARRA. But I've just highlighted a couple of these to note. Within some of the active projects, Drs. Jessica Turnier and Neely have a project that was awarded through CARRA, a large grant in 2022. Analyzing the JDM samples. This was the first request actually to use these samples. And so I know those are in hand, and I know we're actively working on delivering more of these samples to investigators on their team. Additionally, we have a request that is active with Drs. Wahizi and Perfetto. This was also tied to a CARRA Fellows grant and received a lot of interest from our scientific review committee, looking at disease severity and outcomes between African and North American patients. So I know we're all eager to follow the progress on this study. We also have a project by Dr. Wu looking at health equity implications of missing data in the registry, which is a really important topic as we think about diversity, equity, inclusion, and accessibility, and just representation within the registry as well. And so it's an important topic that truly we'll need to address and think of actionable steps to address this and ensure equitable representation in the registry. And then within complete projects, just something to note, a recent publication in the last year or so came out just describing the characteristics of the patients within the CARRA registry. So we won't talk too much about that today, but certainly I think this is a great publication, and we can share the link for this to find out more about baseline characteristics of the registry population. And wrapping up, just looking forward to 2024, some things to note in terms of our focus areas is really ensuring that we have high quality data and samples, focusing on reducing missingness of data and really increasing the availability of data for investigators, and supporting use of these samples, whether that might look like increasing statistical analysis support through CARRA, creating opportunities for RFAs. Still, certainly there's work to be done in this area, but this is one of the key focus areas for our Registry and Biorepository Operations Committee to address. And of course, advancing areas of CARRA's strategic plan is important to ensure financial sustainability and diversification, addressing DEIA initiatives and engaging partners all along the way, whether that be physicians, patients, organizations like CARRA and others, to ensure that we are able to advance research through CARRA. Quickly, some reminders about resources through CARRA. Our grant opportunities are open through March 15th. We have grants that range in sizes from $25,000 for Fellows Awards to the higher amounts for the Career Development Awards. And we also have rolling applications for publication support and data analysis support. So please look into this if you're looking for research funding. And we would certainly encourage you to think about how you can use the registry data or samples within CARRA. We also have JDM committee with many different work groups led by Dr. Susan Kim and Stacy Tarvin on this call, and our data and sample requests are also accepted on a rolling basis, so they don't necessarily need to be tied to the grant process. So please definitely reach out if you have questions about this. And if you reach out, I can connect you with the right individuals internally, but I will pause there for questions.
Thanks, Karin. It's open for questions in the chat box if anybody has any. I think I might kick it off with the foundation partnered with Dr. Tournier and Dr. Neely as part of, and Dr. Deversion as part of the CZI initiative. And one of the things we've been looking at is how to increase diversity in the sample sets that we have and what we can do from a diversity perspective with future clinical trials. So we have a number of programs and initiatives that we're doing, aided by a community advisory board. Just wondering what strategies you have in place to try and increase diversity in your registry and biorepository.
I think that's a great question, Andrew. I think certainly the first step is looking at all of the data that we have in the samples and really seeing if we're able to identify any of these patterns. It's actually one of the things we're actively working on, is how do we increase diversity within everything that we are doing. And so we have had some discussions to start around how we can support some of the sites, for instance, if they might be interested in collecting biosamples and maybe they're great at enrolling patients, but maybe have not collected any samples yet. Identifying if there are maybe any resources that we can provide. For example, we worked with a site a few years ago with a centrifuge program. And so we were able to provide this site with a centrifuge, and they are one of our highest collecting sites. So I think those strategies are going to be tailored to those sites, but certainly it's something we're looking to have a couple of stronger follow-up items in the coming year.